How does the tech that runs our world really work? We break it down to its fundamental truths. This is First Principles.
You're standing in your kitchen holding a small plastic tube. You followed the instructions carefully. No food or drink for 30 minutes. You work up some saliva, spit into the tube up to the fill line, screw on the cap that releases a little bit of stabilizing fluid, shake it, and seal it in the prepaid mailer. You drop it in a mailbox. And then, you wait. A few weeks later, an email dings. Your results are in. You log into a website, and a world of information unfolds. A map of the world, painted with the percentages of your heritage. Northern European, Sub-Saharan African, East Asian. You find long-lost fourth cousins. You learn about your genetic predisposition for caffeine consumption or whether you're likely to have a unibrow. It all feels so seamless, so personal, so magical. From a bit of spit, a machine has somehow reached into the core of your being, into the trillions of cells that make you you, and it has read the master instruction manual. The book of you. It's an act of translation so profound that we rarely stop to question the mechanism behind it. We just accept the result. We assume there must be some kind of impossibly tiny microscope, a nanoscale scanner that just reads the long string of DNA like a barcode at the supermarket. It seems plausible, right? You just look at it and read it. But have you ever stopped to wonder how that actually works? What are the fundamental rules, the first principles, that make it possible to read a biological text that contains 3 billion letters? A text that, if printed out in a standard font, would fill over 1,000 books, yet is coiled into a space 1,000 times smaller than a grain of salt. The truth is, we can't just read it. The real method is far more clever, far more counterintuitive, and frankly, far more beautiful than that. Today, we're opening the black box of modern life itself. We are going to understand how we read the code of life, DNA sequencing. The magic we see today is staggering. It has woven itself into the fabric of our lives so quickly that we've almost forgotten how recently it was pure science fiction. Beyond the ancestry reports and the quirky health traits, this technology is quietly revolutionizing our world. In medicine, doctors are now sequencing the DNA of cancerous tumors. They're reading the genetic misprints that cause the cancer to grow, and then choosing specific drugs that target that exact vulnerability. This isn't just medicine. It's personalized, precision warfare at a cellular level. When a new, terrifying virus emerged in late 2019, scientists were able to sequence its entire genetic code in a matter of days. That sequence was then shared globally over the internet. Because they could read the virus's instruction manual, researchers at companies like Moderna and Pfizer could design an mRNA vaccine in a weekend. Throughout the pandemic, scientists continually sequenced samples from patients around the world, tracking how the virus was mutating in near real time. They were watching evolution happen at the speed of infection, a feat that would have been unthinkable just a decade prior. And the scale, the sheer speed and cheapness of it all, is the real story. The very first time the human genome was sequenced, as part of the monumental Human Genome Project, it was an international effort that took 13 years and cost over $3 billion. $1 for every letter. Today, a machine in a lab can sequence a human genome in a few hours for a few $100s. This radical drop in cost and increase in speed is a technological cascade more dramatic than even Moore's law, which famously predicted the shrinking of computer chips. We've made reading DNA so cheap and easy that we are now sequencing everything. The microbes in the soil, the strange fish in the deep sea, the bacteria in our own guts. We are building a library of all life on Earth, one genetic code at a time. It's an incredible power. but it all rests on solving a very, very hard problem. To understand the genius of the solution, we first have to appreciate the sheer terror of the problem. Let's rewind the clock. By the mid-20th century, we knew the secret of life was a molecule called deoxyribonucleic acid, DNA, thanks to the work of Watson, Crick, and the criminally overlooked Rosalind Franklin. We knew it had a beautiful, elegant structure, the double helix. We knew it was made of 4 chemical building blocks, or bases: adenine, cytosine, guanine, and thymine, A, C, G, and T, and we knew that the specific sequence of these 4 letters was the code that instructed a cell on how to build proteins, how to function, how to be a liver cell, or a neuron, or a skin cell. DNA was the blueprint. But knowing a book exists is very different from being able to read it, and this book, The Human Genome, was of a scale that was almost beyond comprehension. 3 billion letters. Imagine a book containing the word love, repeated 750 million times. That's the length. Now, imagine this book has no chapters, no paragraphs, no sentences, no spaces, and no punctuation. It's just a single, unbroken string of 3 billion A's, C's, G's, and T's. And every single copy of this book is wound up and packed into a microscopic nucleus inside a cell. How on earth do you even begin to read that? This was the challenge that faced the scientists of the late 20th century. The first methods, developed by pioneers like Frederick Sanger, were brilliant but painstakingly slow. They could decipher tiny snippets of DNA, maybe a few hundred letters at a time, through a complex and manual chemical process. Using these early methods to read the entire human genome would have taken centuries, maybe millennia. It was like trying to transcribe an entire encyclopedia by looking at it through a keyhole, one word at a time. The world needed a new paradigm, a way to read this impossible book, not in centuries but in days. And the breakthrough idea, the first principle that changed everything, came from a place of radical, almost nonsensical thinking. If you can't read the book because it's too big, what do? You don't try to build a bigger magnifying glass. You blow the book to bits. This is the moment where we open the box, and the first thing we find inside is a paradox. The key to reading a 3 billion letter string of code is to first, deliberately destroy it. Not just one copy, but millions of copies of it. Let's go back to our analogy of the 1,000-volume encyclopedia. You can't read it, so you take all 1,000 copies and you put them through a paper shredder. A very special kind of shredder, one that tears the pages randomly. What you're left with is a mountain of paper confetti, millions upon millions of tiny strips of paper, each containing just a short random snippet of text. Maybe one says, "He mitochondria is the power hoe." Another says, "House of the cell, producing A," and a third says, "is the powerhouse of the CE." This sounds like madness. You've taken an impossibly hard problem, reading a giant book, and seemingly made it infinitely harder. You've created a chaotic mess of disconnected fragments. How could this possibly be the path to clarity? This is the core misconception we have to get past. Our intuition tells us to read something from beginning to end, in a linear fashion. But when dealing with information at this scale, and at this microscopic level, linear reading is impossible. The solution lies in embracing randomness and parallelism. By breaking the genome into millions of manageable, bite-sized pieces, you create a task that can be tackled all at once. The challenge is no longer reading one giant thing, but instead reading millions of tiny things simultaneously. So now we have our mountain of confetti, our millions of tiny, single-stranded DNA fragments. The next question is the obvious one. How do you read the sequence of letters on each of those tiny fragments? You still can't just look at them. The letters are individual molecules. This is where the second and arguably most brilliant piece of the puzzle comes in. It's a trick of chemistry and light, so elegant it feels like it must be cheating. The principle is called sequencing by synthesis, and the name is the entire secret. We don't read the DNA fragment itself. Instead, we build its other half, its complementary strand, and we watch ourselves do it. Here's the real story. Imagine you're in a dark room. In your hand is one of those tiny, single-stranded DNA fragments. Let's say its sequence is Gattaca. You can't see the letters. Your job is to figure out that sequence. Now you also have a big bucket of loose DNA letters floating around you. A huge supply of A's, C's, G's, and T's. But these aren't just any letters. They have a special property. Each type has been chemically engineered to glow a different color when a light is shined on it. Let's say all the A's glow green, all the C's glow blue, all the G's glow yellow, and all the T's glow red. You also have a helper, a tiny biological machine called a DNA polymerase. This enzyme is nature's master builder. Its only job is to build DNA. It will grab a loose letter from the bucket and attach it to a growing strand of DNA, but only if it's the correct complementary match. Remember, in DNA, A always pairs with T, and C always pairs with G, so you take your invisible strand, Gattaca. You anchor it. The polymerase gets to work. It looks at the first letter on your strand, which is a G. It floats around the bucket, ignoring all the A's, T's, and other G's, until it finds a C, the correct partner. It grabs a blue glowing C and clicks it into place. The moment it clicks, you turn on a laser. Flash. For a split second, that single, newly added C molecule glows a brilliant blue. A sensitive camera records the flash of blue light. A computer connected to the camera makes a note. Position 1 is blue. Blue means C, then a chemical wash is triggered. It does two things. It cleaves off the fluorescent dye, so the C goes dark, and it prepares the strand for the next addition. The process repeats. The polymerase looks at the second letter on your template strand, and it searches the bucket and finds its partner, a T. It grabs a red glowing T and clicks it into place. Flash. The laser fires again. The camera sees red. The computer records, position 2 is red. Red means T wash, repeat. The next letter is a T. The polymerase grabs a green glowing A flash, green. Computer, this goes on letter by letter, flash by flash, color by color, yellow, green, blue, green. After a few hundred cycles, you've built the complete, complementary strand, and you have a record from your computer of the colors it saw in order. Blue, red, green, green, yellow, blue, green. Because you know the color code, you can now deduce the sequence of the strand you just built. CTAAGCA, and because you know the rules of DNA pairing, you can now know, with absolute certainty, the sequence of the original invisible strand you started with, Gattaca. You never read the original DNA. You inferred its sequence by carefully documenting the construction of its mirror image. You sequenced it by synthesizing it. Now here is the leap that makes it all work. You don't just do this for one fragment in a dark room. You do it for hundreds of millions of fragments at the same time. Modern sequencing machines use a small glass slide called a flow cell. This slide is engineered to be a kind of high-density parking garage for DNA. Our millions of shredded fragments are washed over this slide, and they each stick to it in their own little spot. Now, you have a slide with hundreds of millions of different random DNA fragments all anchored in place. The machine then begins the process we just described, for all of them, all at once. In the first cycle, it floods the slide with the polymerase and the glowing letters. A T might snap into place on one fragment, a G on another, and A on a third. The laser sweeps across the entire slide, and the camera takes a high-resolution picture. It captures a field of hundreds of millions of tiny colored dots. a red dot here, a yellow dot there, a green dot over there. The computer records the color at every single coordinate, then the chemical wash. And then cycle 2. The laser flashes, the camera takes another picture of a new pattern of colored dots, and again, for hundreds of cycles. The machine is essentially producing a movie, where each frame tells it the next letter in the sequence for every one of the millions of fragments on the slide. It is a staggering feat of parallel processing, a biological light show that reveals the code of life. So the process of generating the data, the reading part, is done. The chemistry is complete. The light show is over. The machine has produced massive data files containing the sequences of hundreds of millions of short random genetic fragments. Something like GTTAACCG, AATGCCTAG, CCTAGAT, and so on. For millions of lines, this feels like a huge victory, and it is, but it leads us to the final and perhaps most underappreciated part of this entire process. It's the unseen giant upon whose shoulders this entire revolution stands. Because right now, you don't have a genome. You have gibberish. You have the shredded confetti from our encyclopedia. You have successfully read every tiny scrap of paper, but you still have no idea how they fit together. You don't know the story. The final step is not one of biology or chemistry. It's one of pure information science. It is a computational problem of almost unimaginable scale. The job now is to take these hundreds of millions of short sequence reads and assemble them back into the single 3 billion letter continuous string of the original genome. This is the ultimate jigsaw puzzle. Imagine you have 1 million sentences, and your only clue is that they overlap. The end of one sentence fragment is the beginning of another. Your task is to find all the overlaps and reconstruct the original book. Specialized computer algorithms, the workhorses of a field called bioinformatics, are designed to do exactly this. They take a single read, say, Gattaca, and they search through the other hundreds of millions of reads to find others that share a piece of that sequence. They might find a read that goes, CC Gatta. They see the overlap, Gatta, and they stitch them together, CC Gattaca. Then they look for another fragment that overlaps with that new, longer piece, and another, and another. They do this millions of times, building longer and longer continuous stretches of sequence called contigs. It's a brute force process of string matching on a dataset so large it's hard to wrap your head around. This assembly process is the hidden hero of DNA sequencing. Without the massive advances in computing power and the development of these incredibly clever algorithms, the data from the sequencing machine would be useless. It would be a list of words with no grammar, no story. It highlights the profound shift that has happened in biology. The study of life has become inextricably linked with the study of information. A biologist in a sequencing lab today might spend more time writing code and managing data than they do holding a pipette. They are cracking the code of life not just with chemistry, but with computational logic. The secret to reading the book of life wasn't just learning how to see the letters. It was building a machine smart enough to put the shredded pages back together. So let's trace our journey. We started with the simple, almost magical act of spitting in a tube. We assumed some kind of futuristic scanner was just reading our DNA. But when we tore off the lid of the black box, we found a process far more intricate and ingenious. We found that the first principle of reading the impossibly large is to first shatter it into manageable pieces. The second principle was not to read at all, but to rebuild a copy, using a beautiful system of color-coded glowing letters that allow us to watch the process of synthesis, one base at a time, for millions of fragments at once. And finally, we found the unseen giant, the colossal task of computational assembly, of taking the chaotic output of the machine and solving a jigsaw puzzle with hundreds of millions of pieces to reveal the single coherent story of a genome. Understanding this isn't just a piece of trivia. It reframes our entire understanding of what life is. For centuries, we have studied life through the lens of biology and chemistry, of organs and cells and molecules. But sequencing reveals a deeper truth. At its most fundamental level, life is an information technology. DNA is a digital code. It's a four-letter or quaternary system, but it's no different in principle from the binary code of ones and zeros that runs our computers. It's a durable information storage medium of incredible density. And for the first time in the 4 billion year history of that code on this planet, one species, us, has developed the technology to read it. We are living in the first moments of the age of the genome. We are just now starting to read the source code for the entire living world. This ability to read is the necessary first step towards the ability to understand, and perhaps someday, the ability to write. It's a power of awesome and terrifying proportion, to hold in our hands the very blueprint of ourselves and every other living thing. It forces us to ask profound questions about what we will do with this knowledge, what problems we will solve, and what unforeseen consequences we might unleash. We have moved from seeing life as a mysterious, vital force to understanding it as an incredibly complex, information processing machine, and we are just beginning to learn its language.
You've been listening to First Principles. Until next time, stay curious.